At the University of Arizona BIO5 Institute, we are focused on tackling complex challenges such as disease, hunger, water and food safety, and other health and environmental issues facing our families, communities, and the world. BIO5 brings together hundreds of multifaceted experts that include world-class scientists, engineers, physicians, and computational researchers in a team science environment designed to creatively solve difficult problems. This approach has resulted in disease prevention strategies, promising new therapies, innovative diagnostics and devices, and improved food crops. Join us each week as we talk about science with researchers, staff, and students from the University of Arizona's BIO5 Institute.
Hello, everyone, and welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's BIO5 Institute. My name is Sean Caden.
And I'm Brooke Moreno.
And we're here at the BIO5 Institute. We're dedicated to provide hands-on opportunities for the next generation of scientists. This includes high school, undergraduate, and graduate students, as well as postdoctoral trainees. With the support of the state of Arizona's TRIF initiative, we've been able to invest in the success of our postdocs through the annual BIO5 Postdoctoral Fellowship. This fellowship was established by Dr. Michael Johnson in 2019. It has provided monetary awards to exceptional fellows, as well as professional development opportunities to enhance their grant profiles and provide them valuable data in their areas of research. Today, we're talking with Dr. Kate Rhodes, a postdoctoral fellow in Dr. Maggie So's lab and two-time winner of the BIO5 Postdoctoral Fellowship. Dr. Rhodes studies a bacterial strain that causes gonorrhea, a highly prevalent sexual transmitted infection that is most common among 15 to 24-year-olds. Dr. Rhodes, thank you so much for joining us today.
Yeah, thanks for having me.
Yes, two times. So we're going to jump right into the two-time element. So one of the conversations I've had with researchers is how difficult it is to obtain funding. And as a future researcher, a current researcher, you've been able to successfully navigate the awarding process twice. So talk a little, no, very good. So talk a little bit about some of your keys to success in writing a successful proposal.
Well, I think the first key to success for me has been having a fantastic mentor. Maggie knows the ropes, and she is really devoted to making sure that her trainees, get an inside look into how the funding process is, both, internal and external, how those processes work and how to navigate them. So I have to say, she's been an absolutely invaluable resource from that angle, just because, she's basically just downloading all of her years of knowledge directly into my brain. So that's made a huge impact. And one of the things that I think has really kind of helped me, that she's been able to convey to me to figure out this whole process is, try to figure out how to tell a compelling story, and then, simplify things to a goal that you can easily attain in the amount of time that you have. And that's something that has always been a bit of a struggle for me, right? Just because, I think, and that's a problem that I think a lot of scientists have, is that you have this really exciting idea and you see all of the different directions that you can go in. And if you spend all of your time chasing, every single rabbit down every single hole, you're going to have a hard time, reaching, something that's publishable or something that can really move the progress of your research program forward. And so that's been key for me, is figuring out, okay, how do I narrow things down, simplify, and turn that into, something that I can reach in a reasonable amount of time.
And one of the parts of the grant when we're submitting NIH grants that I always find interesting is that you have this specific aims page, which is pretty detailed. You have your research strategy, which is very detailed, but you also have a project summary, which is put it into layperson's terms, 3 or 4 sentences. What are we trying to accomplish? So I think that's a really, really good bit of advice for any future researchers out there.
And it's really difficult to do too. It's a skill that is really, you spend a long time developing it. So.
Well, so I heard find a good mentor and get focused. Those are 2 keys, which I think all of us could, non-researchers alike, could really appreciate. So,
yeah. But that's exciting though. To get funded twice. Tell us about the project that came from your first source of funding and how that ties into your second source of funding coming this year.
Okay, so the first set of funding was training funding to develop this collaboration with a researcher out at the Marine Biology Laboratory at Woods Hole, Dr. Jessica Mark Welch, who has developed this really pioneering technique for looking at microbial communities using some really advanced microscopy methods. And so we were using that to evaluate the behavior or the community structure of this model organism that we've been working with for Neisseria in the context of the gut microbiota.
Okay.
So it kind of ties into, the stuff that I'm going to be working on with the new project, in that, both are really kind of targeted at coming up with new ways of examining bacterial behavior of interest, like our bacteria of interest. So, bacteria in the Neisseria genus, understanding how they interact with the organisms that are in their immediate environment.
Very cool.
Okay, great. Yeah. And you talked about, so your first one, so as it blends into the second one, does it help you get more focused? Or you kind of, or is it the same area of research, or is this?
It's a different way of looking at a similar problem. So in the first project, everything was done on tissues that had been generated from our mouse model that had been colonized with a Neisseria model organism to kind of act as a surrogate for asymptomatic infection, right? But we noticed some really interesting behavioral phenotypes in this bacteria, and we wanted to know, how is this bug interacting with the gut environment, right? The new project is taking, some of those ideas a step further, but looking, instead of looking at an animal surrogate through microscopy, what we're going to be doing is trying to figure out how does Neisseria gonorrhea interact with the organisms of the female reproductive tract. So it's like even one step closer to what would be happening in an actual human body. And the idea with this one is to try to figure out what are the mechanisms that lead to increased susceptibility during, some different disease states that we'll probably talk about later. But yeah, so I mean, they're like I said, they're, kind of interrelated. And that they're both trying to ask questions about the way that Neisseria can manipulate and interact with its environment.
Oof.
And you've used the word microscopy a couple of times. So can you tell somebody who doesn't know what microscopy, I can't even pronounce it, what that entails?
So with this method, so the method that I learned how to use was called Classyfish. So the long title of that is combinatorial labeling and spectral imaging fluorescence in situ hybridization. Sorry, I can't even get it
out.
Wow,
right?
Classyfish. I like Classyfish.
Yeah, there you go. Anyway, so what we're trying to do is use a really advanced microscope that can detect a lot of different wavelengths of light to visualize what's happening directly on the mucosal surface of a tissue sample, using a bunch of different probes or pieces of DNA that are specific to those organisms.
Okay.
And so when we look at that through a microscope, right, we're basically magnifying everything by, hundreds or thousands of times. And that lets us figure out, okay, in the context of everybody else who's a member of this community, what are our specific bacteria doing? Where are they located? Are they forming their own communities? Are they mixing together with these other bugs? What's going on? So it just think blown up image of what's happening on a tissue surface.
Oh, and I imagine there's good and bad forces at play there, right? Because when we think of bacteria, we have this kind of negative connotation about it, right? When in reality, there's good bacteria that really serves us. Can you tell us more about that special balance that occurs in our microbiome, especially in the female reproductive tract?
Yeah, well, and so I think, the idea of everything being either good or bad in itself is kind of an oversimplification, right? Because when you say good or bad, it gets rid of the idea of context. And and to a certain extent, we have to we have to do good research, to do good, in vitro studies. You have to look at one variable at a time. You have to do, reductionist biology to figure out what's going on. But then when you think about that in the context of, okay, it's not just, one kind of bacteria in a test tube, it's, one species of bacteria that may be surrounded by billions of other cells, right? So all of those influences can have an impact on your final outcome. So, I like to think of it, and it's kind of a shift in the field too, to think of it, instead of good and bad bugs, as to think of it as like bacteria with different ranges of pathogenic potential. So that kind of
so we all have pathogenic potential, right?
Well, yeah, because it all, whether or not you get sick, or whether or not you wind up with, an infection may depend on, yes, it depends on the bacteria that you come into contact with, but it also depends on who else is in the neighborhood. It depends on your genetics. It depends on your immune status. It depends on, this whole host of other factors.
Sure.
So you could have two completely different outcomes in two different people from, an interaction with the same species of bacteria, which I think is absolutely fascinating.
Absolutely.
And there's still so much that, and it plays in with the whole idea of this healthy and unhealthy microbiota too. There's still so much that we don't understand, because it is such a complex process, and to develop that microbiota, and it's all of these complex interactions happening together. Like, how do you study like the full gamut of every single variable that could be going on in one human patient? It's fascinating. But yeah, it's a really complex issue.
I mean, to your earlier point about trying to stay focused,
yeah,
when you see all of these different elements, it's hard to say, well, I'm going to have to focus just on this because everything else is, you can't chase everything. And I totally get that.
Yeah, but that's where, kind of looking at things in a lot of different angles becomes really important, right? Because you can answer a piece of a question by one method and another piece of a question by looking at it a slightly different way. And so, hopefully by the end, you can combine all of that to have a better idea about what's actually going on. But yeah, it takes a lot of time.
Absolutely. So we've talked to some of our other researchers about how they got on the path that they're on. So can you talk a little bit about how you chose this path, this particular disease, and how those decisions that you made, that you all of a sudden, like we had one of our virologists on who said, I would never be a virologist. And then she's like, I love viruses, and away she went. Can you talk about your experience with how you decided that path?
I mean, I grew up on a ranch. And so I was always really interested in the vet, like what the vet was doing, whenever, they would come take care of sick animals. And so that was really kind of like my first foray into like thinking about, okay, like what is infectious disease? Like, what does it mean for me? And I think I just kind of kept following that interest. And, as I've gone through my training and all this, school and everything, I think the one thing that's really stood out to me as the most interesting is understanding how these bugs are able to shape their environments. Whether that's, my PhD work was in antimicrobial resistance. So it's like, okay, how do they cope with, the insult of an antibiotic? How are they pumping things out of their cells? How are they, coping with stress? And it's just kind of evolved from there into, the stuff that I'm working on now, this interest, just because I think the ability to ask the question, like, how does this bug cause disease? In the context of what's actually going on in the environment where it's doing those things, exhibiting those behaviors, is really, really cool. And I think to a certain extent, the fact that it is so, like, there are so many different directions that you can go, I talked about needing to be focused, but the fact that there are so many different things going on is something that I think is absolutely fascinating, because in the end, it all kind of ties together.
Right.
Yeah.
And it keeps it interesting. So it's not like, oh, we figured that one out and I've got nowhere to go.
Yeah.
Didn't we hear recently that good science gives more questions, not answers? So,
Right.
and on the note of that, what's your next rabbit hole? What are your next steps in your research with this fellowship award?
Well, so the next steps will be for me to start working with our collaborator, Dr. Melissa Herbst-Kralowitz at UA Med Phoenix. And so, yeah, she and her team have developed this awesome system that lets them evaluate the development of bacterial vaginosis in vitro. And so, the next aim for me is to, go to her lab, learn how they, perform all of their research, learn how they are able to culture some of these like really difficult to culture microbes from clinical samples. Basically,
very cool.
yeah, learn the ropes from her and her team, and then start running experiments. Yeah.
And I think one of the questions that I often have is that how do you find communicating like in public settings? So you're talking about sexually transmitted diseases. We want people to communicate. We know that sometimes there's stigmas associated with sexually transmitted diseases, and how we want people to communicate, especially like today's day and age. We have COVID. We have people who have felt like they couldn't communicate openly about them contracting COVID. I would imagine, we're surrounded by scientists, so we're surrounded by people who want to communicate about these things. How do you find out there in the community you can break down those barriers?
I mean, I think, you're absolutely right. There is, still a stigma kind of associated with talking about sex, and especially sexually transmitted infection. And I think, kind of a broader sense, there's a little bit of a, not necessarily a stigma about talking about women's reproductive health, but there definitely are some disparities there in the way that biomedical research is conducted. It's getting better, but I think the key is to just make things as non-judgmental as possible, because you want people to be able to, like you said, to talk about their own experiences, or to be willing to come forward and seek treatment. And the key to that is to just make the information, unambiguous and approachable, by, making it something that anybody can understand. So yeah, and that's definitely something that I am still figuring out too,
Right.
As a scientist, you get like honed in on this one thing that you think is really cool. And it, you may have all of the background information that's necessary to have it make sense, but you have to figure out how to translate that into giving all of the necessary facts to somebody who doesn't have your background. And so I think that's a major goal for our field, is to do that basically on every aspect of the findings that we have. Is how do we translate that into simple terms that anybody can talk about and feel comfortable talking about.
Absolutely.
I feel like I see that when I see research strategies with all these figures that have all these pretty colors, and it's very bright, and I'm like, somebody understands that. Unfortunately, I do not. So to be able to, that's so critical, to be able to break it down and say, all right, you may not know what you're looking at, but here's what you're looking at. Communication about your science and why it's important, and people in roles like Brooke's and mine. Brooke is part of the Keys internship program, and she's somebody who's teaching science literacy to the next generation of scientists. And our hope is to keep communicating these kind of things out there into the world, and eventually people will get better.
Yeah.
And using your platforms like this to share your research with not just with us and our business office, but the community at large. And thank you for joining us on this platform.
I really do appreciate it. So as you see from our banners behind us, BIO5 is entering its 20th year as an institute. Now, I don't believe you've been with us for 20 years, but talk about your experience with BIO5 and why it is important to you.
I mean, I think the coolest thing about the Institute for me is the fact that there are so many different kinds of research going on under one umbrella. And, especially with the kind of stuff that I've been doing lately, I've had to really kind of expand my tool set. And if you're working in a vacuum and trying to do something new, it can be, you may not have access to somebody who's been doing it for 20 years and, knows all of these tricks of the trade to make your life easier.
Like
And
Maggie.
yeah, like Maggie,
Yeah.
right? Like Maggie, or or just to make sure that the approach that you're taking is the right one, is not going to, lead you astray, so to speak. And so, I think that's why, the setup that we have here is perfect for the kinds of research that we're going to need to, to develop moving forward, where we're pulling in expertise from all of these different disciplines, because, again, like I was saying earlier, the best way to answer a question is to come at it from a bunch of different angles. And that's basically what happens here on a daily basis.
Wow.
Yeah.
So we're doing our job then, because so we've talked a lot about science. Let's talk about non-science for a minute. Tell us something about you that people may not know.
Oh man.
Sorry, I can stall for a little time, but
something that nobody may know.
Interesting hobbies or
I was in marching band for 2 years in high school.
Okay.
Yes.
Yeah.
So are there
I'm a pretty boring person, honestly.
I don't believe that.
No.
So marching band, any correlations with your science, the music, marching band? Not
None.
really.
None,
Not a single
none
one.
whatsoever.
I disagree. The students I work with, there's a heavy correlation between students that come to the Keys program and also participate in marching band. I should look into that more, actually. That said, I've talked to the students about that. I've noticed it with them, and they tell us that it really fosters discipline, motivation, teamwork, collaboration.
I mean, yeah, all of those things definitely come up. Yeah.
Oh,
For
so
me,
thank you to your band teacher.
Yeah. For me, it was mostly you have to be awake at 6 a.m., which
6
I
a.m.
don't do at all anymore, ever. So
Well, it's like if you're awake and actually functional at 6 a.m., you've got a lot on me, because I think I'm awake, but I don't think I'm functional yet.
oh yeah, no, I'm absolutely not functional that early.
Well, thank
Me
you.
neither.
So we never do this, but I'm going to throw it out there. Do you have any questions for us?
I guess what are some topics that you guys have in the pipeline? Like, what are interesting questions that you guys want to ask for, the next set of interviews?
Next set of interviews, we usually go with something that is tailored to the research itself.
Mhm.
But next week, we are interviewing the Keys team.
Okay.
And I am really excited about that, because Brooke is a program coordinator for the Keys team, and she is somebody who's been through the Keys program as a student, and then as a student worker, and then as now a program coordinator, and really making that program run. So I'm looking forward to interviewing them next week and hearing about all the exciting things they have planned for this summer. So that's one thing. And I know COVID seems to be the hot button one, that we have people who have received COVID awards. So we want to hear about how that research is going and what kind of data they're seeing, because we understand it's not a quick process to fix these kind of problems that we're trying to fix. It's a long haul. And maybe we get one or two answers, but that gives us one or two answers closer to solutions to major problems.
Yeah.
Yep.
So there you go. Well, thank you so much for joining us today, Catherine.
Yeah, thanks for having me.
And I said Catherine, but do you go by Kate? Yes.
I go by Kate, but I answer to Catherine. So you're
Well, thank you very much, Kate. Dr. Rhodes, really appreciate you spending the time with us today. And congratulations again on your two successful proposals.
Thank you.
So please join us for future talks, where we will continue to highlight our amazing University of Arizona researchers. To learn more about the BIO5 Institute, please visit our website at bio5.org. And from all of us here at the BIO5 Institute, thank you for joining us.
Thank you, Dr. Rhodes.
Thanks, guys.
To our audience for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the science happening at the University of Arizona's BIO5 Institute.